Differentiation of blood cells.
Differentiation of blood cells refers to the process by which stem cells and progenitor cells develop into different types of blood cells with specialized functions. The differentiation process involves a series of steps, including changes in gene expression, protein synthesis, and cell morphology, which ultimately result in the formation of mature blood cells. Blood cell differentiation is governed by a complex network of signaling molecules like cytokines, growth factors, and hormones. These molecules work together to regulate cell survival, proliferation, and differentiation. Different types of blood cells are produced by different pathways, and each type of cell has its own unique set of regulatory factors that control its development. Red blood cells, erythrocytes. Blood cells differentiate from precursor cells. Red blood cells, erythrocytes, differentiate from the myeloid lineage through erythroblasts. During differentiation, erythroblasts undergo a series of changes, including the loss of their nucleus and the accumulation of hemoglobin, which enables them to transport oxygen. Erythrocyte differentiation is regulated by the hormone erythropoietin, EPO, which stimulates the production of erythrocytes from erythroblast precursor cells. Platelets, thrombocytes. Platelets are formed from large bone marrow cells called megakaryocytes, which differentiate from the myeloid lineage. Megakaryocytes differentiate through endoreduplication, replicating their DNA without cell division, forming large multinucleated cells called giant cells. These cells then release cytoplasmic fragments, which become platelets. Platelet differentiation is regulated by the hormone thrombopoietin, TO, which stimulates the production of megakaryocytes from precursor cells. White blood cells, leukocytes. White blood cells come from myeloid or lymphoid precursors and can change into different types such as granulocytes, monocytes, macrophages, T cells, B cells, and natural killer, NK, cells. Each type of white blood cell has a distinct morphology and function, and their differentiation is regulated by different cytokines and growth factors. For example, granulocyte differentiation is stimulated by granulocyte colony stimulating factor, GCSF, while monocyte differentiation is stimulated by macrophage colony stimulating factor, MCSF. Understanding these pathways is critical for developing new therapies for blood-related disorders and improving the outcomes for patients with these conditions.
Like, comment, and subscribe the channel.